All right. Welcome everybody to the last seminar of the second round of the series. We're so excited to have two speakers today, Dr. Meha and Dr. Abe Stroock, to sort of close our series for this round. So just to remind you, I know the time zone doesn't work for everybody. We have recorded all the seminars for this round of the series, and they're already uploaded on our YouTube channel. So please do check them out. And I think it might be also a great resource for trainees as well, getting into the field of microfluidics and lab-on-a-chip systems. And I will just very briefly advertise for the in-person microTASC conference coming up in October this year in Montreal, Canada. The abstract deadline is coming up in less than 2 weeks on May 14th. So check that out, and it will be great to see some of you in person in Montreal later this year. All right, so without further ado, I will go ahead and introduce our first speaker for today, Dr. Meha. So let me stop share. And I will spotlight Dr. Meha, spotlight. All right, so Dr. Meha is an associate professor in the Department of Pharmaceutics at University of Florida. Her research combines microfluidics and nanotechnology for differentiating, isolating, and loading EVs for precision medicine. She is a senior member in the National Academy of Inventors, a board advisor of the Gordon and Betty Moore Foundation. Dr. He has received many awards, including the NIH Maximizing Investigators Research Award and the Lab-on-a-Chip Emerging Investigator Award. Her research innovation has led to more than 20 pending and issued patents. She is also the founder of Clara Biotech and Exodel. Without further ado, Dr. Meha, is all yours.
Yeah, thank you, Cindy, for the introduction. And I'm so great to have this presentation, especially. And so my PhD supervisor, Jed Harrison, joined the Zoom meeting. I'm really excited. So I want to share our recent research with you, especially focus on this microfluidic device-enabled extracellular vesicle supractical development. Let me see if I can. So I probably want to start with the story, like how this translational impact can be made by the microfluidic approach. If we look at the drug development, that we see for a single drug that finally can be approved by the FDA, which actually take much longer time, usually more than 10 years, and it take a lot of efforts and it spend a lot of money too. So start from the discovery phase, there are more than 50,000 compounds to screen with, and then that can be narrowed down to the preclinical to the 500 compounds, and finally to get the phase 1 and phase 3 trials. That probably limited to 10 compounds, and then at the end to approve by the FDA. So for this entire process, it's very costly and a lot of effort spent there. So each one single drug could be very precious. But how this precious single drug that can go through the clinical trial successfully, it highly depends on the delivery process. Just like we have the drug that we taken in our body, but how that get to the target sites, be able to take a supractical function is still the biggest question in the pharma field. So it's just like the FedEx package. Like when we deliver the package, we want to make sure the package pack the right cargoes and go to the right location. So, but how we confirm that go to the right location? So if we use a FedEx GPS system, so there are apps provided from the FedEx, you actually can track where the package start from and how it goes. And then you can track them until they get to the final destination. So this packaging, tracking, targeting are really the process that determine the precision delivery process. So in the drug delivery field, there are many different modalities, like materials have been discovered for this delivery process to make sure the precision delivery, just like packing the FedEx package to be delivered to your that's to your home address. So here, there's a biomaterials devamp and a synthetic material and a lot of different cargoes that I used. But there's a zero biological treatment in the hearing loss field. It's just because the hearing is even more harder to get a treatment through the delivery process. And the one I want to show you that, I don't know, the slides didn't move. So I want to show you the structure of the inner ear, so which is a complex structure and very small and very sensitive to any treatment. And because it's really embedded tissue, it's very hard to get reached to. So there are many research have been done in the field, try to pack the cargo using different delivery carriers, but because there are quite a lot of biological barrier along the hearing tissue area, and which is hard to reach to the inner ear hair cell. So that's the cell primarily control the hearing ability. And we actually started extracellular vesicle, which could be the very interesting modality to overcome those challenges. So extracellular vesicle are membrane-secreted vesicles. They are very small, around 100 nanometers, and they generate from the cells. So their native derived material is very biocompatible. And through this biogenesis process, they can have the payload loaded into the vesicle membrane structure and then undergo the targeting to the recipient cells. So once the cell receive those cargo loaded and they result into the target cell and perform the outcome and follow biological function. So this process is just like the natural human body FedEx system in our body already. So we try to utilize this extracellular vesicles and for recruiting them to have the payload and working for us. So because those extracellular vesicles, they're not involving any donor cells in the body, they have much longer storage ability and a safe transportation, they have the super biocompatibility and no immunogenicity concern, so very safe to use compared to the cell-based therapy approach. Um, and But in the extracellular vesicle field, there are many challenges right now. The first challenge could be the fundamentally understanding the sorting mechanism and the loading capacity and how we enrich the throughput, be able to have the payload for the supercritical translation, especially for the size diversity, molecular signature is not understood well in the field. So the targeting specificity is not understood yet. So we actually want to step in here using the microfluidic technology. We develop a lot of platforms and try to start the extracellular vesicle that we call it EV toolbox. So in our lab, and we develop many ways for the extracellular vesicle isolation, and we also can do the surface engineering to change their surface property. We also introduced the AI approach to design the peptide for design the EV targeting. And we have 3D culture system for the production line and try to have the much larger production rates that can be clinical compatible. And we have the biogenesis engineering process to engineer the payload. And we also have the cargo loading approach. And we have the CRISPR imaging approach, which using the microfluidic transfection well and to track those EV cargo in the live cells. So, but I think for today, I'm going to focus on this cargo loading approach, which is the good showcase to really demonstrate how the microfluidic technology could have the clinical impact and to help the therapeutical development. So that actually story started from the years ago. I want to just introduce a little bit background of the story. So there's a Dr. Stecker, he is my collaborator. So he actually is a physician from the KU hospital in treating the hearing surgery. But he used the AV, the vector system, the lipid nanoparticle, and those actually cause a lot of toxicity for their patients. And he's really looking for the new modality or the new approach. So we come to the idea, can we pack the CRISPR RNP complex into the extracellular vesicle for the gene editing treatment for the hearing loss. So that moment, I have my postdoc, Dr. Pan, actually joined my team, and she has the background for the CRISPR editing. But so far for the CRISPR editing system, it's still hard to be packed into any delivery vehicle, and there's no any in vivo testing be very effective so far, because this CRISPR editing system, they are unstable in vivo, and also their size is pretty large, because for the case protein and the single guide RNA together, it close to the nanometer range, make it extremely hard as the payload. They're very low loading capacity, and for the in vivo editing efficacy is very low. So we try to have the better solution to address all those challenges. So we actually come with a very straightforward approach using the microfluidic droplets. This, you're probably very familiar with the microfluidics and the fabricate this PDMS, and then you will have the oil phase channel, and with this where the aqueous phase, and then they form the droplets in the microfluidic channel. And we make it as a continuous flow droplet generation, and meanwhile combined with the electroporation system. So we have the electrode actually implemented into this droplet generation system. For each droplet pass through the electric field, then they will form the pulse. And then this pulse where it generates the electric potential across the membrane of extracellular vesicle, and make it open the pore and allow those cargoes to get into the extracellular vesicles. So because it's in the droplet, in the micro scale, as we all know, the mass transfer is very fast, and which allows a fast opening pore, and for the for those cargoes to load it into the EV membrane. And meanwhile, due to the scale of the microfluidic chip, the voltage that you can use is very low. So we just use the simple low power, the DC voltage, like 10 kilowatts, they're good enough for generating this electroporation. But the conventional way, the electroporation actually uses 1,000 volts. So that 1,000 volts actually can generate the heating, the damage on the cell membrane. So, but this low power voltage electroporation makes the very gentle electroporation process, but meanwhile substantially enhance the loading capacity. So after like CRISPR RNA complex loaded into the EV, we can purify and just break the droplets and collect all those CRISPR loaded EVs. So we also can make it as the larger scale production, because it continues to run this droplet generation. So as you can see here, those green fluorescence dots, there are loaded EVs within each droplet, and you can precisely control the speed of the droplet, the size of the droplet, so you can have really consistent production of those electroporation cells and make you in the larger scale. So the conventional transfection is still in the very small scale, and the processing throughput is only around few milliliter. Few milliliter, that's actually not compatible with the clinical usage to develop as a dosage drug. But this droplet generation as a continuous flow, they can generate larger scale of those loaded extracellular vesicles. And we also characterize the cargo loading efficiency with different payload drug. We tested with EGFP. We also have the CRISPR RNP complex and tested with, and we also test the PROTAC drug, which is a oncologic drug to treat the breast cancer, and compare with different loading approach. And our approach actually can produce pretty high loading capacity, especially very suitable for the larger cargoes like CRISPR RNP complex. Here we can reach to the 80% of the loading capacity. And we also characterize the consistency of this loading process using the the qPCR to measure the guard RNA, which loaded into the extracellular vesicles. And with control group, there is no loading payload, and to see the different runs, if there are consistency, so which show quite confident consistency in terms of processing those electroporation process. And interestingly, we find the extracellular vesicle that carrying the CRISPR-RNP complex that actually show really high compatibility. So we compare with the lipid nanoparticles. That's the conventionally used as the drug treatment. Which around like cell viability around 90 to 100% is not bad. But if we use the EV as the carrier to load CRISPR-RNP complex, and the cell viability actually can over 100%, even to 120%, which means this actually promoting the cell and make the cell to be more healthy, more activity. And with this RNP-loaded CRISPR EV, and we test their internalization. So we see the EV-loaded RNP complex actually can penetrate through the cell membrane, getting into the cell nucleus very quickly. So as showing here, the Cas9 protein label, and in the nuclear area, all those Cas9 protein getting into the nuclear area within 1 hour, which is much faster compared to the lipid nanoparticle loaded RNP that's showing here. And the penetration ability into the tissue actually substantially improved as well. This is actually in the mice model. We have this RNP label in the GFP, so which show in the green color, and carried by the extracellular vesicle injected into the mice inner ear. And we can see they're greatly distributed around the inner ear area. So they penetrate really well to getting into the inner ear, and especially uptaken by the outer hair cells and the inner hair cells. Those cells are the major cells that control the hearing ability that we want to target with. The other collaboration actually we started recently with Dr. Stacker and also Dr. Lee, which is the AI faculty recruit to the UF, so which can use a computational approach to predict the peptide sequence that we can engineer on the surface of extracellular vesicles to completely change their targeting ability and their delivery behavior in vivo. So this is the one example that we coated with peptide on the surface of extracellular vesicle, and for treated, especially treated with this peptide coating, and we can see those peptide engineered EV actually only distributed within the inner hair cell region, and also supporting neuron cell here. And compared to the native untreated extracellular vesicle, they distributed everywhere. And this actually helped to localize this delivery to a targeted cell type that we interest for this inner ear hair cell here. So that make the delivery approach to be more targeting, more specific to localize the cell type. In the meantime, we actually collaborate together to find some targets that actually is a controlling factor for this hearing loss. So the MYA7 gene that we found is the major gene mutation that caused the hearing loss. Our collaborator, he actually collect the human paralytic fluid from the inner ear. So the surgical process, then using the needle to get a very small amount of fluid. Because it's only 5 microliter, it's very hard to start it with. But we actually have this immunomagnetic particle we fabricate, which can capture exosomes and using the magnetic field to enrich them. And even in the small amount of extracellular vesicle, only in 5 microliter, we can pull down them to enrich and to harvest those paralytic-derived extracellular vesicles. And we can use to study their concentration, and especially we found a marker that's actually the myosin-A like expressed from those paralytic extracellular vesicle from those human patient sample. And the expression level actually highly relevant to their patient hearing disease status. But from the healthy plasma, it's now expressed. So that means the MYIN7A gene could be very potential targetable gene that control the hearing ability. So we have the CRISPR team and to design this editing system, and we test in vitro and load those CRISPR RNP complex into the extracellular vesicle, and we test their gene editing performance. And we have the different the single guide RNA to screen with, and then to pick the best editing performing guide RNA here, which actually can reach to clear like 100% of the gene editing efficiency in the in vitro system. So that gave us the pretty good confidence to further move into the in vivo treatment development. So we have our collaborator with this unique mouse model, and they can inject this CRISPR-loaded EV into the inner ear structure over here. But the volume that can inject into is only 10, probably like no more than 10 microliter, mostly just 1 microliter, very precious volume, but we couldn't increase the volume because their osmotic pressure difference will cause the hearing loss as well. So we only can use 1 microliter to carry these EVs with the RNP complex and inject it into the scala median that actually can cross this biological barrier to get an inner hair cell, as we're showing here with the confocal image, which had a quite good distribution to the inner hair cell tissue. Compared to the lipid nanoparticle, that actually blocked by the biological barrier, now be able to reach to the target sites. We tested in vivo gene editing efficiency, and after 4 weeks using the NGS and qPCR and the Sangam sequencing, they can further confirm the specificity of the gene editing. We found that with extracellular vesicle loaded RNP, which can give pretty good gene editing efficiency in vivo, but for the because the lipid nanoparticle now be able to get to the inner hair cell, so there's no gene editing actually observed from this in vivo system. We showed the MYIN7A mRNA expression that actually added by our treatment. And we further to continue monitoring this mice model for 6 months and using this auditory brainstem response approach. And we also monitor their gene performance after editing treatment. And we only treat the right ear cells, we only treat the left ear cells and leave the right ear untreated as the control group. And we noticed that after treatment, the left ear actually showed the recovery of the hearing. So you can see this the sensitivity to the sound actually get increased. So we measure the different frequency in terms of response rate to the sound at different frequency. So after treatment, so that's the red line here after treatment, they actually significantly increase their hearing ability and they're pretty close to the healthy status in the green line. So we further test the more animal models and in terms of the treatment response rates and to compare the right ear, which is untreated group, and the treated group from the left ear, and compare with the healthy control group. And we observe pretty close hearing ability to the healthy mice ear and after treatment. So that shows their hearing ability recover. And interestingly, we find the oxidative stress level from the tissue actually highly relevant with their hearing ability, so that we can identify 2 markers we show in the figure here through this confocal staining approach. That 2 markers give the good indication in terms of their oxidative stress level and correlated with the hearing ability, especially at 6 months of the hearing loss mice after treatment. Then there no any oxidative stress markers show up from their tissue after treatment. They give the good supporting information to indicate our gene editing approach actually is functional in the in vivo. So our lab tried to understand the molecular information with extracellular vesicle at precision and control the cargo loading. Especially we can control the surface expression for the in vivo delivery behavior. We actually have this paper posted in the bioarchive, and we still allow the connections, especially from the NIDCD, that I want to acknowledge the Dr. Rona Harsden and also the Lisa. They both actually reached us to our group and want to continue with this collaboration. So we are happy to see our technology actually help the translational impact to the clinical side for them to develop a really precisely controlled gene therapy approach. At the end, I want to give the acknowledgement to the funding support and my lab members, and they're really creative and active young students. And I'm really happy to work with them and to make this work to further push for the future clinical translational effort. I think for now it's 25 minutes, so it's open to the question. I'm very happy to answer any question you may have.
Great. Thank you so much, May. This is so exciting. We have quite a lot of questions. So first is from Julia. Julia, do you want to ask your questions?
Yeah, so I was curious how the microdroplet size influences the exosome loading efficiency. I saw they were 1,000 micron size, and I was wondering how you chose that and optimized the loading efficiency accordingly.
Yeah, so we actually test the different voltage and also the different size of the droplets. So that loading efficiency is mainly controlled by the major two factors. One is the voltage, and the size actually we found is not a major impact in terms of loading efficiency. But the one thing actually will be important and relevant to the droplet size is the throughput. So if we use a larger droplet size, then we can load more per droplet. So then with this continuous flow, that means the production rate, the processing throughput will be significantly increased.
Okay, great. We have a
Thank you.
question from Prima. Prima, would you like to ask your question directly?
Yes, thank you so much, Sarmia. That is like really exciting. So I was just wondering if like the EV size would matter in this case, like, and what is the size range that you would say be the best in the efficiency, in terms of efficiency? Like, is it like less than 50 nanometers, less than 100 nanometers, or like larger than 100 nanometers? And in terms of delivery side of it.
Yeah, that's a good question. So usually we don't want that too small. So we have our isolation approach developed a few years ago. So we have pretty good control in terms of size of the extracellular vesicle that we isolate from. So the size we try to control around 150 nanometer to the 200 nanometer. So which have the better room for accommodating more RNP complex. So definitely we don't want that too small, below 50 nanometer. So 150 to 200 that we use.
Okay, thank you.
Great. We have a question from Sai. Sai, do you want to unmute?
Hi, yes. Thank you so much for the exciting talk. Briefly, you had mentioned like the system of particles called nanopalms that were used to capture the exosome. I was wondering if you can elaborate on that. It seems like an exciting way to capture and keep them intact.
Yeah, so that paper we published before to the Nature Communication Biology. If you're interested, I can share a paper to you. So basically, which is the nanopalm structure, have this pom-pom-like surface property. Those nanographin sheath layer actually anchored on the surface of magnetic beads, increase the larger surface volume that we can immobilize the antibody on those surface and to increase the capture capacity. So that capture probe that we use is antibody. So that antibody can be customized. Usually we use the generic marker like a CD9 or CD81 CD63 for the generic EV capture. That can be customized to some tumor marker like AbCam as well, if we want to pull down very specific population of the EV. I think for this work, now we use a generic marker to pull down the EV, especially from this human paralytic fluid from our collaborator. The very nice part using those magnetic particles, because they can be aggregate, enriched to the small volume. It will not dilute sample. The most clinical sample only in like 5 microliter, very small volume, and we don't want to lose any of them. We don't want to have dilution introduced. So we use those magnetic particle to capture, and there's a photocleave linker actually anchored to the antibody. So after capture, then we shine the light, so the light actually breaks the bond and to release the captured exosome after capturing and washing steps. So we can collect those intact exosome at the end, either to study their surface marker or either to discover their internal marker.
Great. So we are at the 5:30 Pacific time mark. So there's still a lot of questions for May. And May, if you don't mind staying after the seminar for maybe 5 to 10 more minutes, I think we can go over the other questions. Thank you so much again.
Yeah, I will stay here. Yeah.
Okay, great.
No problem.
Thank
I'm going to
you.
stop here.
All right, so we will now transition to our second talk and see if I can find Abe and spotlight him. Okay, that's light. Okay, all right. So I will now introduce Dr. Abe Stroock. Dr. Abe Stroock is a professor in the Smith School of Chemical and Biomolecular Engineering and adjunct professor in the School of Integrative Plant Sciences at Cornell University. He is the director of the NSF Center for Research on Programmable Plant Systems. The Stroock Lab focuses on manipulating and measuring dynamics and chemical processes on micrometer scales, with an emphasis on biophysical contexts. Current efforts center around the transport, thermodynamics, and physiology of water in plants, and in synthetic systems inspired by plant function. All right, without further ado, Abe, it's all yours.
Thank you, Cindy, and everyone of the organizers for this opportunity. So I'm going to tell you a little bit about the ideas in the Center for Research on Programmable Plant Systems, and a long technical story. I'll tell it briefly, but along from my lab of how we got to this place of Internet of Living Things with plants. So this is a story that started actually with my first PhD student, Tobias Wheeler, and ran with a number of excellent students and postdocs over more than a decade now, and been a collaboration with a number of excellent plant biologists and plant physiologists. So the Center for Research on Programmable Plant Systems is a science and technology center of the NSF, and it unites an unusual diversity of life scientists, engineers, computer science, and social sciences, and even in just the plant sciences, from very technological plant transformation through plant molecular and cell biology, through to translation toward the selection, breeding, and genetics of the food that the crops that feed us. So the vision of a programmable plant, my favorite, comes from a colleague here at Cornell, Margaret Frank, in plant biology, and it is a vision of communication, where our current collaboration with plants, our current ability to interact with plants, is limited to the point where we have very primitive ways of managing the plant life around us. And so the vision that Margaret illustrates here is one in which we endow the plant with the ability to communicate its inner biology and the physics, chemistry, and living systems in the world around it. to us in a form that we can capture, we can see, we can digitize, and we can inform the plant of things that it doesn't have access to, as simple as the weather forecast. So this ability to gain efficient connections to the inner workings of a plant system opens onto an Internet of Things idea, embedded systems, where we could start to place these living systems in connection with multi-scale modeling, digital twins, ideas that we have very much in industrial contexts. And in fact, very much I'm living on an Internet of Living Things with my cell phone and my smartwatch. But a focus of the center is really to build this node on the Internet of Living Things, which is a sophisticated set of interfaces with the living plant in its microenvironment and its ecology. So why? Well, plants are important. And we cover a major portion of our land mass with managed plant life, but we do it in a manner that is open loop. And in the context of water, 70% of the water used by humans is used in agriculture, and about half of it is wasted. Even in our famously wet Northeast here, this is a growing challenge for the well-being of our population, if not just the economics of the agricultural system. Nitrogen's the same story, but even worse. Plants only utilize about a third of the nitrogen inputs into the plant. And again, this is open loop, uninformed by the internal status of the plant. So you can't control what you cannot measure, and that is a big challenge. And one of my reasons to be happy to present here is because it's a challenge for people like us, like you. And closing the loop is a measurement challenge at the biological interface in the environmental context. So another less visible reason to be involved in this need for engineers and scientists like you is that we have a huge R&D effort that's continuously churning out new plants to improve yields, to improve nutrition, to improve resilience. But here's an example of the lines that are the green revolution lines of increasing yield through the 20th century. And what you see is in many environments, these are plateauing. And one of the reasons is measurement, insufficient measurements. Measurements here is using a modern tool to measure a very macroscopic coarse property of the plant, here, the leaf angle. And so plant breeding is done with enormously sophisticated genetics and bioinformatics, but extremely primitive measurement of plant properties, leaving an enormous black box of biology in between that significantly limits how we can deal with big challenges like efficiency of use of water and nitrogen. So one inspiring thing for me in this area was something that a famous plant geneticist, Ed Buckler here at Cornell, said to me. He says, I can find the genes associated with any trait. And it took me a little while digging to find out that he meant any trait I could measure 10,000 times in the field. And so that, though, is inspiring. But today, the tools are very primitive. And what I show here on the right is a big, heavy instrument that is used to measure the internal status of water stress in plants. So finally, why crops? Why this center? Why I'm excited to speak to you is because people like us have been neglecting plant life. And I know there's lots of beautiful problems in the biomedical sciences and in chemistry, but plant life is in need of us, and we are certainly dependent on it. And so just a stark example is that we do not have the equivalent of even a hypodermic needle in the context of plant biology. So I'm going to turn to kind of my origin story in this area, which is really around water, water stress, and a curiosity. What started as a curiosity about the physiology, the plant physiology of water stress. But water stress is important, and anyone who has to think about whether to mow a lawn knows that without water, there's no growth. But without water, it impacts an eventual yield. It impacts in subtler ways with optimal levels of stress, quality, in particular of fruit crops like wine grapes. And too much water is not good, leading to, for example, disease. So what is water stress? Well, this was our question. The plant physiological textbook will say it's tension, negative pressure in the liquid within the vessels, the microfluidic vessels called xylem that carry water from the soil to the sites of evaporation in the leaves or needles. This is an exotic state of liquid. It's not prohibited physically, but it's a fragile one. It's using liquid like a rope, and like a rope under excessive tension, it can break. And in the context of a liquid rope, that is boiling. And so what is this, how is this structure captured? And it turns out I'm going to just give you a little mapping onto an idealization of what the xylem structure is. And at its base, what the physiology says is that it's a bidisperse porous medium, or a bidisperse microfluidic structure, where we have what I'll tell you, what the state of stress, which is essentially the mechanical pressure. I'll use psi in these liquid paths is negative absolute pressures. And so this, by Young-Laplace law, says that somewhere in the system we need to generate that stress. And in particular, we need to generate that stress in equilibrium with a dry world. And so there's two length scales that are critical. One is a nanoscale structure, and one is a microscale structure. And the microscale structure is to allow for macroscopic transport at high permeability, and the nanoscopic scale is in order to hold on to this, allow for nanoscopic menisci that can generate very large stresses. So just for some of you, this may help. For others, it may not. But really, what a plant is very much living on the desorption isotherm of an unusual porous medium, what has been called in the literature an ink bottle porous medium, where you have a body of the bottle and a throat of the bottle, two length scales of pores. And what the plant is exploiting is the liquid branch of the adsorption isotherms of this material. And if you think of it, if you think these adsorption isotherms are really an expression of the cubic van der Waals-like equation of state, liquid is being pulled into tension along the liquid branch as it comes to equilibrium with a vapor that is becoming unsaturated. And another cut of the thermodynamic state space is that this is into a superheated state where the liquid wants to boil, an isothermal trajectory into superheat under tension. So the trouble with water stress has been that it's very hard to measure. And so I'm showing you again what is a 30, 40, 50 pound instrument that is literally a pressure, a gas-filled pressure chamber at tens of atmospheres of positive pressure. And this crazy poor physiologist is having to put his eye right up next to this pressure chamber. So this is made that we don't close the loop on water stress in agriculture. So this was a curiosity to me and my first graduate student, Tobias Wheeler. Is it really true that these vessels are holding stress? And we built the most primitive possible microfluidic mimic of the xylem structure, where in this case, The nanoscale structure was the structure of a hydrogel, and the microscale structure was microfluidic channels formed directly within that gel. And so it turned out that we convinced ourselves, absolutely, the plant sciences have the physics right. The materials are available to allow for a structure to hold tension. And in fact, in this first structure, comparing here to the negative pressures assessed using that pressure chamber I showed across all different plants in different climates and ecologies, this synthetic structure was holding on to tensions several fold higher than had been documented in true plants. It was fun. About 10 years after we did this work, some Australians in the outback found a warty xeria where they came to this very same threshold for the stress in water. I'll just mention that this threshold of 200 some atmospheres of tension is not the predicted fundamental breaking strength of liquid water, which is well over 1,000 atmospheres. But in our hands and in a number of other experiments, and including this warty xeria, it seems to be the practical strength of liquid water for unknown reasons. So we had fun with this plant of xylem-inspired structures. Here we're watching segmented in the same way. Here's little spherical segments coupled to each other by nanoporous structure. What we're watching is the release of tension, the boiling events in this confined ink bottle structure. And this has interesting poroelastic dynamics coupled to the kinetics of homogeneous or heterogeneous nucleation of the vapor phase. We could use these stresses, and in the context of few nanometer pores, we can generate many hundreds of atmospheres of stable liquid stress in nanoscopic pores. And this allows us to probe at the limit of the continuum limit of just a few nanometers. And what we find is that, in fact, all of continuum properties, both thermodynamic, interfacial properties like the effective surface tension, Young-Laplace, they all held across multiple simple liquids. The only violation of the continuum limit was the effective absorption of one layer of molecules. A fun fact is that when you have a pore liquid that's able to come to equilibrium with a dry atmosphere, you put it under tension and it changes the thermodynamics of dissolution. And in fact, at around 50% relative humidity, the solubility of oxygen in the pore liquid nearly or more than doubles relative to ambient liquid. And we can have fun with the architecture here, starting to more approach the architectures of true xylem, the long microscale conduits separated by nanoporous connections, and start to look at the effective medium behavior and how we can tune the permeability and stability of these moving around liquids under tension. So we were having fun with these with the thermodynamics and transport of these xylem-inspired materials when a colleague, Alan Laxo, a plant physiologist, came and said, I think this could be more useful than that. And the reason was because he spent his career carrying around that pressure chamber trying to do stress physiology in plants. And so during the same period, we were developing, we quickly moved out of this, what was quite a terrible hydrogel to work in, into a silicon architecture where this is a classic diaphragm piezo resistive pressure sensor on one side of the chip. And on the back side of the chip, we have a cavity that is our test cavity for the liquid we're going to put under tension and surrounded by porous medium. It turned out that in the end, we actually had to go quite far towards the mimicry of the xylem structure because the pocket of liquid, the kind of connectivity to the outside world where we want to measure stress, that conductance was too low without some sort of vascular structure or xylem-like structure give us high axial conductance. And so this really is, again, this architecture we call a microtensiometer is really a variant on this ink bottle where we have our cavity of the test liquid connected to the outside world by this bidisperse porous structure. So this is a classic MEMS process. There are a few extra steps. But what's been most exciting is that here our mimic of xylem becomes an implantable structure, an implantable sensor in woody species. And this is a system that is now commercialized by a former student, Michael Santiago, who really brought the MEMS system all the way to its first implantation in plants at FluoroPulse based in Davis, California. And so we literally place our little synthetic xylem structure in contact with the active xylem of the plant, and it gives us back through that MEMS pressure sensor a measurement of the stress. And so this is now over the last 7-8 years started to be something we can do in a lot of species. You'll see that heavily there the fruit and nut woody perennials, although we have had some success in some annual plants like maize. So this is the beginning for me of the Internet of Living Things. It's a MEMS-based way of connecting a living plant out in the field to an electrical signal that we can send to a cell tower. So this gives it just a sense of the variety of dynamics we can see. I called this the pulse of plants, and the company is called FluoroPulse. And what the pulse is the daily dynamics of the plant opening its stomatal pores and its leaves up to capture carbon dioxide and inevitably losing water. As it loses water, it pulls the whole tissue into tension, tens of atmospheres on a daily basis. In Ithaca, New York, midsummer, we have rainy days, wet, sunny days. You see this highly heterogeneous dynamics driven by atmospheric conditions. And in fact, you can start to see that the clouds coming back and forth across the sun drive a dynamics on the minutes to hours time scale. In a commercial orchard in the Central Valley in California, the dynamics is very distinct and entirely driven by the pattern of irrigation here. And what we see is, in fact, there's I'll show in a minute, just very nonlinear dynamics in the plant as it responds to the atmosphere. And this was a nice example of seeing a whole year where in an orchard in a vineyard in Davis, where you see large stress during the summer, you see the loss of leaves, you still see dynamics through the winter, and then you see the beginning of development of the canopy again and the emergence of stress. So this, some excellent students and postdocs really brought this out into the world and started for us to build our little Internet of Things here on Potted Apple. And I'll show you just a few ways in which now the plant itself is our microfluidic system. And so I'll just show you dynamics in a little bit more detail of this is the the transpiration rate day by day. You see this is a cloudy, wet day, very low transpiration. And what you see here is the mirror in the stress pattern. Here, in contrast, it's hot and sunny every day through the summer in the northern Central Valley of California. And here you see the very distinct dynamics, not at all mirroring the rate of the transpiration. It turns out that if we do a simple Ohm's law on the stress and the flow, the driving force, the change in pressure across the system, and the evaporative demand, what we see is very simple behavior in the wet world of apple growing in New York State. Really, this is Ohm's law. On the other hand, if we're in California, something else is going on here. And in fact, what we see is we have this very nonlinear response where the flux is the same every single day, but the conductance of this whole plant system with its soil, plant, atmosphere system is changing radically even as the conditions stay the same. And so that just gives you the beginning of a flavor of how plants actually in the full organism, in the full field context, can start to be a clean transport system for interesting study, understudied by people like you and me. So in the context of our wet world here in New York, where we have this, really, we just have a constant conductor, we can do very well by taking the weather, wind speed, this is relative humidity, temperature. We can do very well predicting from an initial condition over long trajectories. We can predict the dynamics. So we can now start to be able to tell management of a crop what the weather forecast is, and think about how to do, for example, model predictive control. And in fact, we've done some of that with our data with a colleague in systems engineering, Funchi Yu, here at Cornell. So that's a story of how plants are served as our inspiration, and now more and more are subject of inquiry, and an area where there is translational opportunity that not many people like you are taking advantage of. And I'll just say that what's very exciting for us now is to be able to collaborate with people like Margaret Frank, where the tools start to be by having a living system as our system of interest. We can start to use molecular tools, genetic tools, to define, to do new measurements of biophysical processes, and to manipulate those biophysical properties at the molecular scale. So thank you very much. And I'll be happy to answer any questions now and for the next few minutes.
Thank you so much, Abe. This is fascinating. Okay, we have questions. I also have a lot of questions. First question from Sai. Sai, do you want to unmute and ask your question?
Hi, thank you for such an interesting talk. I was wondering if the sensor, the microtensiometer, can be implemented into any plant, or are there any requirements on the plant type, or any requirements into the types of plants that the sensor can be implemented in?
So it really targeted woody perennials like this apple tree that we're looking at, and that is the most mature context in which tight water management is done. If you've been in the Central Valley, you'll know that almost all irrigation is done with drip and micro irrigation. So it's a context where water is precious, and the outcomes are very strongly dependent on water stress. So that's been our area of interest. We've developed since a tool that allows us to go into, in a very different way, into leafy annual plants, which is a nanoparticle that we bring into the vapor spaces inside of the leaf, and gives us an optical readout of this same parameter, this water potential. But so yes, the short answer is it's woody perennial crops that are best targeted with this microtensiometer.
Thank you.
Great. We have a second question from Julia.
Yes. So yeah. So
yeah,
sorry,
thanks for this interesting talk and introducing us to this. Oh, sorry. Can you hear me?
Yeah, I can hear you. Go ahead and ask your question.
Okay. Yeah, for this interesting area with plants and all we can do by measuring them and communicating with them. I'm curious whether the water stress can also be an indicator that a plant is suffering either from sort of plant disease or other kind of infestation, whether the sensors could be useful for kind of monitoring the health of the plant, and individual plants could be treated earlier in the case of some sort of infestation or disease.
So that's an interesting question, and it's one I got to say I haven't had before. There is a huge focus on plant pathogen management. It's whole department in schools of plant science. Yes, there's a coupling because, in fact, the devastating diseases ruining citrus in Florida and olive in the Mediterranean are xylem-dwelling microbes. So there is a connection, and undoubtedly one of the ways in which they are impacting health is through stress. But it's not an application context that I have that we've considered. But yes, I think the answer is under some circumstances, yes.
Thank you. So we are at 6 o'clock Pacific time, and we have more questions for Abe and also for May. If you don't mind staying for another 5 to 10 minutes, and I think we can go over the questions. But for now, I will officially close the seminar series for this round. And I'd like to thank both speakers again and everybody for joining today. And we'll stay in touch, and I guess we'll be posting announcement about the next round. Thank you, everybody, and Abe and May, if you don't mind staying.
Yeah, no problem.
Good.
Right. I will stop the recording, and then we can go into more informal Q&A. Thank you.